What are stem cells? Stem cells have specific aspects that make them different from other types of cells. The first characteristic is that these cells are unspecialized that can divide to produce more undifferentiated stem cells. Another aspect is that these cells can be manipulated to become other types of cells. There are three main types of stem cells: embryonic, adult, and iPS. The first type of stem cell is embryonic. These types of stem cells are able to remain undifferentiated until needed. If they are able to form clumps, the result could be rapid differentiation into a number of different types of cells. This type of cell can also be determined by the type of medium the stem cells are on. Embryonic stem cells are taken from excess fertilized embryos that are obtained from clinics after in vitro fertilization. These embryos are donated for research. The embryonic stem cells are taken from the inner mass of the blastocyst. Cells are then transferred from the embryo to a culture dish with a medium on it. The cells then divide and cover the culture dish. There is also a feeder layer of cells that help the dividing stem cells to attach to the dish as well as provide nutrients. In order to maximize production of embryonic stem cells, they are often removed from the original culture dish to a new one where they continue dividing. This can be repeated many times to produce millions of cells. In order for these cells to differentiate into specific roles, certain transcription factors must be present in the undifferentiated cells. Specifically, the OCT4 and NANOG transcription factors allow embryonic development and the differentiation of these embryonic stem cells. OCT4 assists in regulating genes that affect multiple types of cellular function. NANOG works to sustain pluripotency in embryonic stem cells. The next type of stem cell is an adult stem cell. These undifferentiated stem cells are found in existing tissues like the brain, teeth, heart, and bone marrow. Adult stem cells maintain and repair the tissues in which they are found. These stem cells can be grown in the lab and manipulated to become specific cells, but their capacity to divide can be limited. Some examples of situations where adult stem cells are used could be using bone marrow stem cells to regenerate bone, creating cells that are capable of producing insulin to treat type 1 diabetes, or using stem cells from cardiac muscle to repair it following a heart attack. However, removing these types of stem cells from tissue limits the amount of times they are able to divide. Some experiments are being done to fix this by growing adult stem cells in a culture that can be differentiated to produce specific types of cells. Induced pluripotent stem cells are cells that began as an adult stem cell but were genetically transformed to behave more like an embryonic stem cell. These are taken from tissues and are manipulated to where they are able to become a variety of cells similar to embryonic stem cells. One difference is that the tissues created from iPSCs have a very low chance of being rejected by a person's immune system. This is because the iPSC cells are going to be extremely similar to the tissues that produced them. Another unique aspect about iPSCs is that they behave like embryonic cells that can give rise to many types of cells, but it removes the controversy surrounding the use of embryos. iPS cells are also being used for research on human diseases and provide a source of human tissue to be used in experiments. iPSC cells start as an adult stem cell that is taken from an organism's tissues or organs. A virus is then used to insert reprogramming factors, including OCT4, into the adult stem cell, causing it to become an iPSC that behaves more like an embryonic cell. These cells could become cells of skin, muscle, bone, nervous system, or organs.
Stem cells offer an incredible way to treat many diseases. With their ability to regenerate and differentiate, they can be used to treat diabetes, heart disease, spinal cord injuries, vision problems, and many, many more. Despite being discovered over 30 years ago, they are even useful to treat diseases of the blood, bones, and skin. So how exactly can stem cells be used in the medical field? Well, in this video, we will discuss how stem cells can be applied to cancer, umbilical cord blood, and bone marrow. Let's start with cancer. Cancer is one of the most detrimental diseases to have, from the emotional toll on the patient and their loved ones to the painful treatments. However, stem cells can often be used to avoid high doses of chemotherapy and radiation in order to battle cancerous cells. During the stem cell treatment of the cancer patient, stem cells are used to replace the cells that were damaged during chemotherapy or radiation. With this replacement, stem cells can then renew the bone marrow's ability to produce more blood cells. There are also treatments called autologous transplants, where stem cells are obtained from cancer patients before they start chemotherapy, and then put back into the patient's body following chemotherapy. Another application of stem cells includes umbilical cord blood. Obtained from the umbilical cord after birth, stem cells from cord blood samples are often stored and frozen for future therapeutic use. Unlike bone marrow transplants, cord blood does not have to be closely matched to the recipient in order for the transplant to be successful. Cord blood is also very useful because of its multipotency, meaning it can differentiate into a variety of cell types and tissues. Finally, cord blood is used as an inexpensive, readily available treatment plan, as cord blood stem cells can also be obtained from the placenta, the umbilical cord vein, and the umbilical cord matrix. Finally, let's talk about bone marrow stem cells. We mentioned before that bone marrow requires a close match between the donor and the recipient. Coming from human blood, hematopoietic cells have many properties that are useful in stem cell transplants. First, they are able to make copies of cells with the same or similar characteristics as the parent cell. In recent studies, these cells were able to produce copies to last many mice lifetimes. Imagine how useful that would be to the human body. Another useful property includes their ability to differentiate. However, this differentiation requires many factors and substances to do so. Third, hematopoietic cells have the ability to migrate to different parts of the body. This can be useful in a case where a patient presents with multiple ailments throughout the body, which could be treated with these stem cells. Lastly, bone marrow stem cells have the ability to induce their own death. You might be thinking, why would that be useful? Well, in the event that something goes wrong, whether it be in replication or cell division, bone marrow stem cells have the ability to conduct apoptosis, just like with our own somatic cells. In order to properly function in the human body, stem cells must differentiate into specific roles throughout various systems. To do this, stem cells respond to internal signals that are controlled by genes along strands of DNA. These internal signals are the cell's instructions for all its functions and structures within the cell itself. In addition, stem cells can also respond to external signals that include secreted chemicals from neighboring cells, contact with neighboring cells, and exogenous molecules. These interactions with internal and external signals allow the stem cell's DNA to differentiate. In this process, the DNA develops epigenetic marks that can restrict DNA expression in the cell, which is then passed on through cell division.
Today we're going to talk to Dr. Marrow about his work with stem cell research. He received his PhD from Johns Hopkins and is now a professor here at NC State in anatomy. Hi, Dr. Marrow. Thank you for joining us today. So why do you think stem cells are important?
They have many potential uses in human medicine. We can use stem cells to repair tissue and regenerate parts of the body when your body can't. A study conducted here at NC State created a cell-mimicking particle. It is believed that these cell-mimicking particles can act similarly to real cardiac stem cells. These cell-mimicking microparticles are more stable when stored, and due to the fact that they aren't really cells, do not elicit an immune reaction. This therapy aims to be a less harmful way to treat cardiac tissue injuries and rebuild muscle. For example, these particles do not undergo mitosis and therefore cannot create tumors.
What are some common misconceptions people have about stem cell research?
Some people believe that stem cell research is illegal, but this is not true. No. In fact, the National Institute of Health gave almost $1.3 billion towards the research into stem cells in 2013. Another controversial topic is the use of embryonic stem cells. They have additional uses when compared to stem cells in that they are more versatile and can be differentiated. The embryos used do not come from aborted fetuses. Currently, the procedures we use to extract the cells leave the embryos inviable after. There are other extraction methods and sources of stem cells we are searching for. For example, stem cells from pre-implantation genetic diagnosis, which were found to have a genetic problem, could be used since they will not be reimplanted. Some people also believe that stem cell research can eventually lead to cloning. There are multiple groups such as the National Academy of Sciences and the International Society for Stem Cell Research that have restrictions to prohibit cloning.
What is some current research you're excited about?
I am really excited about somatic nuclear cell transfer. This method replaces the haploid material of an unfertilized egg cell with the diploid material from the nucleus of a somatic cell. This will grow into an immortal cell line of the chosen cell type. So, if someone needs neurons, this new diploid cell could be told to express itself as a neuron. This technology is particularly exciting because the use of embryonic stem cells and the ethical debate behind them may be phased out in the years to come.
Thanks for meeting with me. I look forward to hearing about your further work with stem cells.
Human stem cells, and more specifically human embryonic stem cells, are a very controversial topic. There are two sides to the debate, those who support and those who oppose stem cell research. These two sides can be further classified into the duty to prevent and alleviate suffering and the duty to respect the value of human life. The following discussion will present the two sides of this argument.
The main opposition to human stem cell research is the use of embryonic cells. This is a particularly well-conversed topic because of the harvesting of the cells kills the embryo, essentially ending a life. So not only do you have the issue of embryonic stem cell use, but you also have the issue of deciding when life starts. Does it start at conception or does it start at birth? Many people who identify as pro-life are against embryonic stem cell research because the embryo is no longer viable after the cells have been harvested. There is also a religious aspect to this debate regarding the protection of human life. Another negative aspect of using stem cells is that the stem cell industry is often used in scamming desperate individuals. It gives people unrealistic hope and can end up harming the health of an individual more than helping them.
The other side to the debate is that the collective that believe in the duty to prevent or alleviate suffering. It should be known that most embryonic stem cells come from unwanted embryos used by fertility clinics. No life is harmed. The possibilities that surround stem cells are virtually endless because stem cells can become any type of human cell. Heart cells could be made to help heal someone with heart disease. People with degenerative diseases like Parkinson's could potentially be cured, or at least their suffering lessened. Stem cells also contribute to researchers gaining more knowledge about diseases. So a question to ask is, does the possible life of an embryo outweigh the life and well-being of a living person?
The information above has to do with the social/ethical aspect of the debate. But what about the legal side? What about federal funding?
The fight for federal funding began in 1996 when the Dickey-Wicker Amendment was passed. This amendment banned the use of federal funds for the creation of a human embryo or embryos for research purposes, or research in which a human embryo or embryos are destroyed, discarded, or knowingly subjected to risk of injury or death greater than that allowed for research on fetuses in utero. In 2001, President George W. Bush issued an executive order prohibiting the use of federal funding using any embryonic stem cell lines derived after August 9, 2001. In 2005 and 2007, Congress tried to pass a bill extending funding for stem cell research, but the bills were vetoed both times by President Bush.
President Obama reversed the 2001 executive order in 2009. The U.S. District Court in D.C. ruled in favor of embryonic stem cell research in Shirley v. Ceballos in 2011. This ruling continued to allow funding for embryonic stem cell research. In 2000, the NIH released the following guidelines for the use of stem cells in research. Private funds must be used to obtain frozen embryos from fertility clinics. The embryos must have been created for fertility purposes only, and the embryos must be in excess of the donor's need, and the embryos must be obtained with consent of the donor.
Both sides have been presented, but the topic continues to become more complex as we find out more about stem cells and how they can be used in human medicine.
Hey, my name's Deoxy, and I'm going to talk to you today about genetics and stem cell research. An understanding in genetics is important to the field of stem cell research. We have to have an understanding of the mitotic division in embryonic stem cells and how that plays out in the body. Additionally, we use genetics to understand how these cells specialize and differentiate based on the presence of transcription factors. Stem cells can also further the field of genetics. For example, Parkinson's is a known genetic disorder that causes neural degeneration. One possible treatment would be to use stem cells to reproduce the neurons to regain lost brain function. Without the underlying genetic knowledge about Parkinson's, stem cell therapy wouldn't be as useful because we wouldn't know what cells to replace. So why does stem cell research matter? Stem cell research is an exciting field with the potential to help a lot of people. The opportunities that stem cells provide are as infinite and diverse as they are, and could greatly improve the quality of life.
I'm Dr. Marrow,
And I'm Deoxy.
and thank you for learning about stem cells with us.